Hello, my name is Gavin Douglas, and I'm an assistant professor in the Department of Biology at the University of New Brunswick in Fredericton, New Brunswick. And today I'm going to give a redo of a lecture I gave back at the end of October at the University of Western Ontario with a few minor updates. The recording at that seminar didn't work, and so I thought I would just redo it. And I'm also preparing to give this lecture again, so it's a good opportunity to do so. But before I jump into the presentation, I first wanted to just go over, really just introduce my research program, which I just started at the University of New Brunswick back in July 2025. And so here's the campus pictured alongside the beautiful St. John River. My research program is really motivated by this huge growth in the number of bacterial genomes in recent years. So this is a breakdown of the number of genomes that have been archived online in various databases from 2010 to 2024. And what you can see is at this earliest time point, there was about 7,000 genomes that were archived. And now, at least as of 2024, there was just about 550,000. So there's been an enormous increase in the number of genomes that are out there that can be analyzed. And so what that really means is that microbiology, which is and should remain primarily an experimental science, can be greatly complemented by a data science component of trying to dig into these genomes that are becoming available to make inferences that are useful in their own right, as well as those that complement experiments. One really interesting observation that I'm excited to dig into more is this idea that there's enormous strain variation in nature. And so a recurrent observation is really represented by this Venn diagram on the left, where we can see this is just a replotting of a classic finding from 2002 of 3 E. coli strains, which were highly pathogenic and also highly divergent. And so you can see here just 40% of the genes were shared by all 3 strains, whereas in some cases, up to 22% of all the genes found across them were found just in this one strain specifically. And so a lot of strain-specific genes, at least in this small example, but it turns out in general, when you look across diverse species, you tend to see this strain variation, which is often referred to as pangenome diversity. And so this U-shaped plot on the right is fairly representative of what you'd commonly see, especially for organisms with so-called open pangenomes, meaning that there's a high degree of diversity and divergence between strains. And so on the x-axis here is just a representation of the number of genomes that encode each gene, and then the y-axis is the percentage of all genes found across the pangenome, the so-called set of all genes across these genomes. And so you can see a large proportion is found in only one genome out of 10 here. And so this is just a toy example, but it's pretty representative of what is typically seen. There's a lot of interesting questions about what's driving this diversity across strains. One important explanation is that this must, to some degree, be due to horizontal gene transfer. So just as a really quick reminder, the 3 primary mechanisms driving transfer between cells, rather than vertically from mother to daughter cells over generations, are transformation, the uptake of free DNA from the environment, conjugation, plasmid transfer between cells, and transduction, so virus-mediated transfer of DNA between cells. So getting back to my research program, I'm going to be focusing on 3 main pillars. The first is bacterial population genomics, and the idea is that my group will be contributing to publicly available resources that characterize bacterial genome diversity, as well as those that characterize elements across genomes. For real focus on pseudogenes, these are degenerating elements, which I have recently shown appear to be a really useful tool for digging into this question of gene distributions across different strains of the same species. So it's a really useful tool in general for digging into evolutionary dynamics. And so that's something I think is understudied in bacteria, especially in this context. And so it's something I'm excited to dig into more. And then the holy grail, but I want my group to get at eventually, is to develop a framework for distinguishing neutral and selective factors that drive strain diversity. So that's the overarching question I'm most excited about. But there's a lot of other interesting questions that I'm very open to collaborators and new trainees to come work on as well. And so the other major pillar is phylogenomic bioinformatic tools. And so I have a background in developing a number of tools for microbiome data analysis. And so a couple examples of new tools I want to continue working on are those that can be used to identify co-occurring genes across strains, so those that putatively are co-evolving together and could be functionally linked, while also controlling for phylogenetic relationship and things like location in the genome. I also want to build a framework to use this information from existing genomes to better assess genomes from mixed sequencing, so from metagenomic sequencing, as I'll talk about. So there's a lot of information out there that's really underused in terms of how we're treating new sequences we're getting from the environment, basically. So I hypothesize there's a lot of interesting ways we could use our existing genomes better. And then lastly, much like I was just mentioning, I have an interest in looking at genomes in the natural environment, specifically strain variation. And so I'm working with a number of collaborators looking at genome diversity across natural bacterial strains in ocean samples, as I'll talk about today, but also in lake samples and even the honeybee gut microbiome to some degree, which I won't talk about today, but I'd be happy to if anyone wants to reach out with any questions. And then lastly, I thought I would just mention, in case anyone's interested, I have a growing interest in meta research. So basically research about how research is done. And one representative of the project there is looking at citation accuracy across the microbiome literature. So basically looking at high throughput citations across papers and then looking at, is the claim that was actually cited, is that well supported or not? And trying to characterize that across a large number of manuscripts. So that's a project I hope to be working on soon. If that's the sort of thing that interests you, please feel free to reach out. So getting to today's presentation, it really falls under this third pillar, looking at strain variation in natural environments. So there's been a lot of work in the past to dig into the factors that drive the rate of horizontal gene transfer across different bacterial lineages. So this has been well characterized as soon as a decent number of bacterial genomes became available. Folks were digging into looking at similar genes across distantly related organisms and saying, hey, this looks like horizontal gene transfer, and building networks of which lineages appear to undergo the most horizontal gene transfer. So you can see some so-called highways of transmission between major classes under Proteobacteria here, or Pseudomonodonta, as it's called now. I apologize. And a major recurrent finding is that organisms that share a niche are more likely to undergo horizontal gene transfer. So this has been shown in a number of different investigations. And by a niche, I mean that there's similar environmental conditions, or it's literally co-localization together. So I think this observation makes a lot of sense. But what remains unclear is, okay, but what can we conclude from this? What other factors could be involved here that could explain this association beyond simply this nice interpretation of, okay, if you share a niche, you undergo horizontal gene transfer together? And one really important confounder to think about is phylogenetic distance. And the reason for that is pretty clear once you see an example. So if you think about different bacterial cells in two different environments, so the green ones being environment 1, blue in environment 2, if we identify that it looks like the cells within each environment, they tend to undergo more horizontal gene transfer together. So these could be two different species, for instance, in these environments. It's not uncommon to find that those species that undergo more horizontal gene transfer, they're actually more closely related as well. So it just turns out that organisms that are more closely related often are in more similar environments and co-occur more often as well. And so there's also this idea of genetic distance is mechanistically linked to how easily one can undergo horizontal gene transfer. So recombination, for instance, is limited by genetic divergence. And so a certain level of similarity is needed for that to occur, which is often needed for horizontally transferred DNA to actually incorporate into the genome, for instance, and other mechanisms like compatibility with plasmid systems and with bacteriophages, for instance. So these can also be this is a really important factor in terms of all of these effects that are associated with phylogenetic distance that can modulate horizontal gene transfer. And so disentangling the relative importance of co-occurrence and phylogenetic distance isn't trivial. So you might be wondering, well, surely co-occurrence is associated with horizontal gene transfer to some degree, right? Because physical proximity is required. But I just wanted to point out that that's not necessarily true, at least when we're talking about co-occurrence of extant organisms. So there are other possibilities when we're thinking about organisms today compared to their ancestors which underwent this horizontal gene transfer event. So yes, co-occurrence, co-localization, so physical proximity must have occurred in the past to some degree for a transfer event to occur between ancestral cells, but this could have been transient, or the ancestors their descendants may no longer co-occur, and so we might not see the same pattern jump out in data today. It's possible that the inferred transfer finding may, although these, as I'll talk about, are highly similar genes across different organisms, they may have been indirect and mediated by other microbes in a larger network. And so the actual relationship, again, the exact transfer event between the ancestors of those genomes we're looking at today, there wasn't even direct between those. And so hopefully that's clear. And it's another idea which I think may be a little counterintuitive to folks who haven't worked in microbial ecology before, is that there might be widespread opportunities for horizontal gene transfer. And this is related to the idea of, that's just really a common saying that was popularized many years before microbial environmental sequencing became popular, but really became more prominent after that, which is that everything is everywhere, but the environment selects. And so this idea that there is the potential for at least a high proportion of the diversity of microbes to be in any environment, but it just depends on the environmental conditions. And so I think there's a lot of caveats underlying that, but it's related to this idea that there might not be that many barriers to genes being transferred across diverse environments. But it just really depends on the selection acting on those genes mediated by specific environmental conditions. And so in that degree, it's the environmental conditions and not the requirement of certainly extant organisms co-occurring today that's driving the probability of a gene being found in a certain environment and driven by horizontal gene transfer. And so for those reasons, it's not necessarily true that we expect to see a clear signal of co-occurrence between organisms that have or are within the same horizontal gene transfer network, have genes that are highly similar and mediated by horizontal gene transfer to some degree. Okay, so getting back to the overall question of what factors are associated with co-occurrence and horizontal gene transfer, this was investigated in a recent paper in Nature, Ecology, and Evolution, which really motivated a lot of our own work. So it was published in 2024, and I'll just go over one of the main findings. So these findings can be a little confusing the first time you take a look, so I'll go through slowly. The idea is that they looked at a large number of operational taxonomic units, so OTUs, based on 16S ribosomal RNA gene sequences. So basically, markered barcodes that are found across all bacteria. You can just sequence that gene or a fragment of that gene and get an approximate idea, usually of the genus, the family level of that organism, sometimes lower, but usually the genus. And basically, you can get an idea of how taxonomic units within that genus, or that at least that genus itself, are distributed across different environments. So they looked at a vast number of these marker gene, these 16S sequencing data sets, and looked at how these different OTUs co-occur. So again, these coarse units of a taxon, so not a specific organism necessarily, but a grouping that all have this same fragment of the 16S gene, and how they're distributed across different environments. And then they also looked at genomes in a database that matched this 16S sequence and looked at what genes were in that genome, and then looked at putative horizontal gene transfer events across genomes in that database. So in other words, genes that looked like they were actually transferred between them due to using a phylogenetic approach. And so what you can find here is that the number of genes transferred, so each dot here is another operational taxonomic unit. So this is focused on one OTU, and each dot here is another OTU for which it looks like at least one gene was transferred. And if you look at the correlation between genes transferred and co-occurrence, there is a strong Spearman correlation. So for instance, this would be another OTU where it looks like about 10 genes were transferred to this OTU between OTU1 and this one, and their co-occurrence score is about 0.25. So just give you an idea of what the data is again. The problem is, if you look at, again, on the y-axis, the number of genes transferred and their distance between OTUs in a tree, so the phylogenetic distance, they're negatively correlated. So this is, again, related to this confounder I mentioned of phylogenetic distance being highly associated with horizontal gene transfer as well. In this case, negatively associated. It's also negatively associated with co-occurrence, as I mentioned. And so there's a major confounder here of saying, well, we can't just trivially interpret this as being a causal relationship between co-occurrence and genes transferred, because there are these other possible confounders as well, in this case, phylogenetic distance. And so the authors, what they did was they regressed out the signal of phylogenetic distance. So if this might be a little confusing, but the key idea is that they fit a model to the data of co-occurrence versus phylogenetic distance, and then they looked at the residuals, so the distance from the fit line to the actual points, and then they did their analysis on these residuals rather than on co-occurrence directly. So rather than on co-occurrence directly, they looked at the residuals after fitting a model to co-occurrence versus phylogenetic distance. So again, if that's confusing, the key point to understand is that they basically corrected for phylogenetic distance. And long story short, they did find that there was still an association between horizontal gene transfer and co-occurrence after this correction, but it was weaker than before. And so they showed this across a high number of OTUs. So just under 4,000 OTUs, which they could do this comparison. You can see that post-correction in dark orange is still higher than the background. And so this was, I think, a great example of a broad-scale data analysis on microbial ecology. But a key limitation which the authors recognize is that this is based on 16S ribosomal RNA gene data. So they linked it to reference genomes, but the actual co-occurrence data is based on this very coarse data. And also they didn't, because they focused on such a broad set of samples, they didn't dig into specific questions like, does it look like it's physical proximity that's more important, or niche specific adaptations that's driving this link between co-occurrence? And so that really motivated our own project here. So I already kind of mentioned this, but 16S ribosomal RNA gene sequencing, for anyone not familiar, just very briefly, the idea is that you're sequencing into this highly conserved gene and getting something like a barcode of what organism is present, so usually the genera or species and their relative abundance in each sample. And this is in contrast to shotgun metagenomic sequencing, where the idea is that you sequence all the DNA in a sample, again, from any part of the genome. And this can allow you to identify species as well as, if you have enough data, the strains that are present across samples, and to assemble the reads into genomes, metagenomic assembled genomes, and to also assess the community's functional potential in that way, because you're identifying the genes in these assembled genomes rather than just getting a barcode and then looking at what genes are in the genome database. So, getting back to our question here, so we were motivated by this study that looked at this again, the association between co-occurrence and phylogenetic distance, co-occurrence and horizontal gene transfer after correcting for phylogenetic distance. And we wanted to ask, can we recapitulate this signal in metagenomics data specifically, and focused on a single environment? So we decided to look at ocean samples because there's been numerous metagenome assembled genomes produced from ocean samples for which metagenomic sequencing has been applied, particularly the Terra Oceans project and the GeoTraces project as well. And there's also been a lot of work collating and producing consistent metadata across ocean samples for which there's metagenomic sequencing. And so it really allows us to dig into more detail to try to recapitulate the signal of independent data, but also dig into more detail environmental data and how it's associated with signals of horizontal gene transfer as well. So our main hypothesis was that we would still see this positive association between co-occurrence and the rate of horizontal gene transfer across these ocean microbial genomes, even after controlling for phylogenetic distance. So very much so following up on this previous study with new data, and again, a new angle in that we can dig into environmental parameters to a different degree as well, as I'll talk about. So these other confounders, depending on how you look at them, or interesting variables to investigate as well. So we started out by analyzing just over 15,000 previously assembled, mainly metagenome assembled genomes that were from the Terra Oceans sampling expeditions where metagenomic sequencing was done. And you can see the breakdown of the phyla that we analyzed in the right column here. So you can see the major phyla represented. So mainly bacterial with some archaea as well. And so across these genomes, we identified putative horizontal gene transfer events. So this was based on just under 57 million gene annotations across these genomes. And what we did was we clustered all these genes using CD-HIT at 95% identity over more than 95% of the length of each gene to produce 20,792,000 gene clusters. So again, these were clusters of genes, sometimes of only one gene, but of one or more genes where all those elements, those different individual genes, were 95% identical or higher. And then we aligned these genes within each cluster using MAFT and computed the Hamming distance to get the actual exact number of nucleotides that differed between each gene cluster, and identified the bidirectional best hits within each cluster. And the idea here is that we wanted to identify those that were found on genomes across different genera. So basically, these are highly similar genes in terms of their nucleotide similarity, but found across highly divergent lineages, which we would not expect due to basically common descent. So if they were basically orthologs, but were actually transferred through horizontal gene transfer. And so through this approach, we identified just over 9,000 putative horizontal gene transfer hits, which are the focal set we focused on. But this was actually, although this was the main approach we focused on in this work, we tried a few different approaches as well, because it's really important to think about how approaches could be impacted by the choice of bioinformatics workflow. And so we did do a sensitivity analysis to make sure our results were robust. So this is just an overview of the horizontal gene transfer patterns based on this focal set of putative events we identified. So here you can see that there was the majority of these events are occurring between genomes that differ at the genus level. So on the x-axis here is the taxonomic level the genomes differ at. So the first column here being that the genomes differ at the genus level, the family level, and so on. And so we did identify some that actually differed between bacteria and archaea as well, but mainly at lower levels. And this last row here is the actual number of genomes compared in each case, which is important to control for, because otherwise it might look like there's actually more going on at the phylum, but that's just because there's more phylum level comparisons being done. And so basically another way to look at this is just the number of events divided by genome comparisons here on a log 10 scale. And you can see that there's this gradual decrease at higher taxonomic levels of fewer horizontal gene transfer events inferred, as you'd expect. And so this gets back to this idea of phylogenetic distance being related to the rate of horizontal gene transfer as well. And so this was definitely expected. And we initially binned the transfer events based on whether they were highly similar versus more distant, which gives us some gauge of how ancient the events were, and we saw similar patterns for both. But actually, most of the events we identified were greater than 99% identical. So we first wanted to ask, mainly as a sanity check, just to make sure we were interpreting this data correctly, of what type of genes are enriched among these hits. So how we investigated what functions these genes were enriched for was to look at what COG category each gene was found within, and then seeing whether they were enriched for certain categories. So COG stands for clusters of orthologous genes, and it's one way of looking at how, basically the broad functional grouping of a gene. And so in this case, COG categories are split into groupings like translation-related, cell motility, etc. And because there's been a lot of work previously looking at what genes are horizontally transferred, we have some expectations about which ones are depleted versus enriched, as well as some that just have unclear expectations. So these groupings here where there's been some contrasting findings in the past. And so the way we framed this analysis was to look at genes that were called as horizontally transferred at both more distant levels as well as more similar, as well as at different taxonomic levels as well. So this is the taxonomic level that the genomes differ at. And then to see whether they were either depleted or enriched for horizontally transferred genes at each of these levels. And so as expected for translation-related genes, for instance, in general, what you find is that these are depleted among transfer events, whereas things involved in the mobile, for instance, so including a lot of mobile genetic elements, prophages, transposons, that sort of thing, those are highly enriched for transfer events, which makes a lot of sense, given that would be the mechanism in many cases that'd be driving horizontal gene transfer. So in general, what we found is that the categories, at least at the genus level, usually were in the expected direction. So this is especially true for more distant events where all the expected depletions and expected enrichments were in the expected directions. It was a little more mixed at more similar identities, and especially at higher taxonomic levels, which, as a reminder, were the minority of transfer events. And so overall, we can conclude that these transfer events make sense a lot, especially for these more distant ones in terms of expectations from the literature. But it also highlights that you can get some different stories depending on how you divide the data. So it's especially interesting to think about how the signal from lower levels, which would be the majority of horizontal gene transfer events, can cover up more subtle effects that's occurring between basically genes that are transferred between very taxonomically dissimilar taxa as well. So what I've been talking about now before is just characterizing putative horizontal gene transfer events. So to get into this question of how co-occurrence is related to that, we needed to actually look at metagenomics data. And so as I alluded to before, we took advantage of publicly available data. So 1,865 ocean metagenomic samples, which we mapped reads against these over 15,000 metagenome assembled genomes. And actually, this is not just metagenome assembled genomes, but majority of them are, but just genomes, it should say. So that was with cover M, and looked at the presence-absence profile of these genomes across these samples, and then used a number of genome co-occurrence metrics to look at how they're co-occurring. And so again, the idea there was to look at a few different approaches to make sure that our results were robust to just whatever workflow we chose. So similarly, we also wanted to look at phylogenetic distances, which we did by computing trees based on universal genes from all 15,339 genomes, and looked at the distance in terms of branch length distance in this tree across all genomes. So this gives us a continuous measure of phylogenetic distance. And so with all these data points, these data types in hand, the co-occurrence, phylogenetic distance, and horizontal gene transfer, we wanted to first assess, can we find the similar associations as previously identified? But a focus here on genome pairs, so pairwise comparisons of genomes. So we did find this clear association between co-occurrence, phylogenetic distance, and horizontal gene transfer. So first, just to focus on HGT versus co-occurrence, we found that, again, that organisms that do co-occur, they tend to undergo higher rates of horizontal gene transfer. So you can see that clearly in this contingency table. So again, most organisms do not co-occur, most genomes do not co-occur in the data set, but most of those, a higher proportion of those that do co-occur also undergo horizontal gene transfer. In addition, there's also a very clear signal of higher phylogenetic distance for genomes that do not show evidence of horizontal gene transfer. So again, similar to before, we showed you that genomes from different domains, for instance, have less evidence for horizontal gene transfer compared to those that just differ between different genera, so more closely related ones. So essentially, if you just compare the two panels here in the left and right, and just looked at the average of the box plots in each panel, there's clearly a lower phylogenetic distance for genome pairs that do show evidence of horizontal gene transfer. So that's what this panel on the right is. All of these genome pairs have evidence of horizontal gene transfer, whereas these do not. The y-axis is phylogenetic distance. So you can see, again, the phylogenetic distance on average is much lower on this side, on the right side. Interestingly, there is some modulation with co-occurrence as well, which I'd be happy to talk about if anyone's interested. Basically, I think this is due to what's called collider bias, where genome pairs that both co-occur and show evidence of horizontal gene transfer, on average, tend to have slightly higher phylogenetic distance compared to those that do not co-occur but have evidence of horizontal gene transfer. So again, that's a little counterintuitive, but the main take-home message is that we find the expected signals between these three variables. In addition to these three variables, we also had access to, for this subset of the analyses, these coarse variables related to things like depth, latitude, salinity, oxygen. So related to the sample, of the actual metadata of where the ocean samples were taken from. So in addition to the metagenomics of the samples, other measures were taken as well in many cases. So because for this component, we were doing a very broad meta-analysis across these very diverse data sets, there's not that many environmental variables that could be compared. I'll get back to other environmental variables for a more specific subset of samples as well, which I think is more interesting. But nonetheless, even based on these coarse variables, you can see a really clear signal of genome pairs that do not undergo horizontal gene transfer also having differing much more for all of these variables. So a higher y-axis here means that they're more different for each of these variables. So for instance, they have less similar oxygen levels if they do not undergo horizontal gene transfer, whereas they're more similar on average if they do show evidence of horizontal gene transfer. So this is based on what metagenomic samples these genomes are found across. So this is basically another example of potential confounders that could be more causally related to horizontal gene transfer levels, but that are confounded with things like co-occurrence and phylogenetic distance potentially as well. So these are variables we also wanted to control for when doing our analysis. And another potential and variable of interest we were interested in was what was the water size fraction of the sample. So how was the water filtered prior to sequencing for particles of different sizes? So in ocean ecology work, it's common to filter samples to 3 microns, less than 3 microns, to get free living samples. So that restricts larger particles, which could have multiple bacteria attached to them. And so there was 906 such samples we were able to parse out from the data set we're analyzing. And about 846 samples that include both free-living and particle-attached bacteria. So the reason I'm using that, they're saying both free-living and particle-attached, because basically it's a broad range. It's not specifically particle-attached, but it includes particle-attached bacteria due to it just being a broader range of less than 20 microns. And so for this analysis, we identified genomes that were specifically associated with each of these sample sets. So either specifically associated with free living samples or with this broader set here. And because I know that's a little confusing, I'm just going to give another little explanation of that. So these two categories are the free living samples and the free living and particle attached. And the idea here is that you can, the free living and particle attached includes basically the entire range of sizes. So both free living and larger particles, whereas again, this is just small particles, primarily free living. And because this is a very diverse range of data sets we analyze, this is the exact breakdown of the filter sizes used to filter the water samples that we use to define the free living and particle attached grouping. So NA meaning there was no filter used for the lower or the upper filters that were used. So again, these are diverse samples, but in general, the main difference here is that this is a much more inclusive grouping, whereas these are much more targeted towards free living organisms. I have to turn the pointer off again. Right, so I've given an overview of a number of different data types, different variables we're interested in exploring here. And this is just kind of a monstrous workflow diagram showing all of these different variables we're interested in. And so the main takeaway from this slide is that we wanted to take all of these pieces of information, the horizontal gene transfer, pairwise phylogenetic distance, whether they were associated with these broad, less filtered, broader size fraction samples or more specific small particles enriched for free living, how the genomes co-occur, and how environmentally similar the genomes are in terms of the samples they're found across, and put all of this information into a single table of all the genome pairs, and then to run logistic regression to test which of these variables are associated with horizontal gene transfer. So the idea is that this will implicitly control for other variables in the model and help control for this problem of confounding variables or all these co-association between variables, and really identify which variables are most significantly associated and what their effect sizes are. And so this plot summarizes the coefficients from that logistic regression model. On the y-axis are all the tested variables. And remember, the response here was, in this case, is where the genome pairs have at least one instance of horizontal gene transfer identified. So it's a yes, no of whether there's horizontal gene transfer. And these are all of the variables used as predictors in the model. So co-occurrence, free living and particle attached. So again, that's related to whether the genomes are in that pair, whether they're both associated with metagenomic samples that are either, again, in those size fractions like I just described, the phylogenetic distance of the genome pair, and also the environmental distance based on all of these factors here. And then again, so what's shown is the coefficient in terms of log odds of each of these variables. They're all significant here except for oxygen. And what you can find, basically, the way to interpret this is that negative values mean that the variable is negatively associated with horizontal gene transfer. Positive means it's positively associated. So one of the main take-home messages here is that co-occurrence is significantly associated and strongly associated with horizontal gene transfer while controlling for these other factors. So we cannot simply reject that co-occurrence is important among extant genomes to explain horizontal gene transfer given these other confounders. And also phylogenetic distance is clearly associated, much more so than these environmental distance factors. So that was an interesting notion as well. And then lastly, we found this noisy signal of genome pairs that are both in these less filtered samples, which include both free living and particle attached bacteria. They show a stronger signal of HGT. And so I'll get back to that in just one moment. So based on this result, we had a few follow-up questions. The first is, which environmental factors are most associated with horizontal gene transfer? So again, we looked at really broad factors in that analysis I was just explaining, but can we look at more specific factors that really seem more interpretable in terms of their relation to horizontal gene transfer? We found that there was this weak association between horizontal gene transfer and genomes in these less filtered samples. By that I mean those that include both free living and particle attached bacteria. But if we look at specifically large size fraction samples that are enrich specifically for particle attach and exclude free living? Do we still see this signal? That would be interesting, because that our hypothesis there was that particle attached bacteria might have higher rates of horizontal gene transfer because they are in closer physical proximity of other bacteria on that particle. And so what we wanted to do was dig into these questions on individual samples rather than taking this genome pairwise approach, which was useful for a really broad scale analysis, but for interpreting the importance of other variables besides co-occurrence, a little tricky. And so we looked at this measure of horizontal gene transfer prevalence. And so we define this here as the proportion of genomes in a sample with at least one gene called as a horizontal gene transfer event. So again, so you can think about this as across different metagenomic samples, different numbers of genomes in each sample will show evidence of horizontal gene transfer. And so this is a way of looking at the proportion of each of those samples. We also looked at different subsets of the samples as well, rather than just the broad set of all samples. So we first looked at 131 samples, specifically from the Terra Oceans project, which again, for our small particle enriched, so free living samples. And we did this because, first of all, it reduces variability. It's all from the same sampling expedition, the same data set. They also have a lot of data related to the environment for these samples in particular. And so we focused on these focal variables, which you can see on the y-axis here, clustered across these samples. So you can see there are some strong associations between some of these variables. So in particular, there's things like chlorophyll A, which is a measure of phytoplankton biomass, PAR, photosynthetically available radiation, is highly associated negatively with depth, for instance, but not redundantly so. It's not exactly the same information. And so we did want to include all of these variables to see whether there was a clear association between them and horizontal gene transfer prevalence. And so based on the marginal associations, so the individual associations between variables and HGT prevalence, we could find a number of clear associations. And so one I'll just point out is that this association between PAR, again, photosynthetically available radiation, and HGT prevalence is really clear. And there's a very clear separation between those with basically no PAR, so no light, and those that do. So the low light ones have much higher HGT prevalence compared to those with high PAR, which was something that really jumped out from looking at these data. But this is just based on the individual associations. And the whole point here is that we wanted to look at all of these variables together to see which ones were more important after controlling for the effects of others. And whether we also expect we know these variables are highly related, so we wanted to correct for non-linear associations and interactions between these variables as well. And so the way we did that was by using a random forest model to predict in this way as the response variable HGT prevalence and use these environmental variables as the predictors. And so sometimes this is described as a black box machine learning classifier. It's actually less of a black box compared to most machine learning, at least more sophisticated machine learning tools. But nonetheless, it's difficult to interpret compared to things like linear regression. But it can capture nonlinear dependencies between predictors and is a very powerful tool. So I'm not going to go into great detail here, but the idea is that you can you can get the general idea if you pause and take a look at this and want to look up random forest on your own. But the idea is that you can take your input data, your predictors, and on a subset of the data where you have what the answer is, whether in this case you should play golf today or not. That's the predictor. It's a binary value. You can build a number of different trees based on different subsets of that data to figure out what split of the data. So in this case, let's say you take a cutoff of how sunny it is, and then decide yes or no, play golf. But you can see there's other possibilities in this decision tree where it's associated with other variables, and this allows there to be these dependencies and finding the exact cutoffs. But because you're only using a subset of the data and you're making so many of these decision trees that make up the forest, that basically makes it really robust to variation across different subsets, and it forces it to be more generalizable rather than overtrained on just your specific data set. So basically, it's a way to make a powerful model in a very quick way that's fairly interpretable compared to other machine learning approaches. So based on this approach, we identified key environmental variables associated with horizontal gene transfer, including chlorophyll A, PAR, and this measure related to turbidity in the water, as well as these others that are mentioned here. And so all of the colored bars are those that are significant in the model based on permutation-based variable importance, so how important they are in the model. So this was interesting, and I'd be happy to talk more about this. So one key takeaway here is that variables associated with depth, although actually interestingly depth itself was not one of the main variables, but things like phytoplankton biomass and photosynthetically available radiation, even to some degree particular organic carbon levels, which are highly associated with depth, in this case negatively associated, they're also highly negatively associated with horizontal gene transfer. And so long story short, one of the key things to jump out of this data was that depth, or at least variables associated with depth, are strongly positively associated in terms of the direction of increasing depth with horizontal gene transfer, which is interesting because this is also something that came up in another recent paper showing that there's markers of mobile genetic elements that are seen to be more abundant at lower depth as well. So this is on the same lines as that, actually, and it was encouraging to see. So we also wanted to perform this analysis, this follow-up analysis focused on samples specifically that contain free-living bacteria versus those that specifically contain particle-attached bacteria, or at least that were filtered to be highly enriched for each of those sets. And do those show clear differences in horizontal gene transfer? So to do that, we did not have these specifically particle-attached bacteria in our initial meta-analysis. Those are normally used for more of the protist fraction when you're looking at sequencing data rather than the bacterial fraction. And so what we did is we acquired 160 additional metagenomes that were publicly available and to perform this analysis. And so although the focus is generally on the protists in those samples, they are also enriched for particle-attached bacteria. And we are able to identify 43 biological samples where separately there was these small versus large size fraction filtering done, which allowed us to do this paired comparison of the metagenomic sequencing. And so again, just to be clear, I explained these two categories before of size fractions, but now I'm talking about particle attached specifically. So those are those that samples that include this wider range of sizes that specifically exclude the small size fraction, so exclude the size range of free living bacteria. You can see the exact filter cutoffs there. So what was really clear from this comparison was that there is a very clear difference in terms of the horizontal gene transfer prevalence across these groupings. So on the y-axis is the HGT prevalence. So again, the proportion of genomes of at least one instance per sample. So each dot here is a different metagenomic sample. And the two groupings here, again, are these metagenomic samples split by size fraction. And you can see a very clear increase, or I shouldn't say increase, but higher level of HGT prevalence in the particle-attached group compared to the free-living group, as well as increases in a number of elements related to mobile genetic elements. So I'd be happy to talk more about this, but the pro-MGE, for instance, refers to matches to a database of mainly transposases and recombinases. So genes that are enriched for, or genes that are annotated as having those functions. And so basically this is saying the proportion of genomes in each sample that encode at least one gene that matches that. So you can see a lot of them do, but nonetheless, there's a higher proportion in the particle attached, as well as genes that are associated with plasmids, associated with viruses, or have a provirus. So all of these were significantly higher for the particle enriched group, except for scaffolds encoding a provirus. So overall, this is consistent with there being higher rates of horizontal gene transfer in particle attached bacteria, at least in those metagenomic samples. So to conclude, We found evidence for widespread horizontal gene transfer across the surveyed genomes, including at higher taxonomic levels. We also found genome co-occurrence across the ocean samples to be significantly associated with horizontal gene transfer even after correcting for phylogenetic similarity, and also after correcting for these broad environmental factors as well. And when zooming in on more specific environmental factors, we found an association between higher HGT and factors associated with increased depth. So in this case, these are factors negatively associated with depth, but photosynthetically available radiation and chlorophyll A, which are very low at high depths, and really were where we saw the highest levels of HGT, in addition to higher nutrient availability as well, which was associated with HGT. And we also found a really clear signal of higher HGT in terms of the prevalence in the samples, as well as HGT-related elements, so mobile genetic elements, specifically in particle-attached bacteria, or at least the metagenomic samples with that size fraction of particle-attached bacteria. So this work was just published yesterday, actually, in the ISME journal. And so you can see the full author list here. So we're very excited to finally see this out. And I'd like to thank all of my co-authors in this project, especially Nico, Samuel, and Jesse, who really drove this project forward, and my funding sources over the last couple of years when I was working on this. And of course, the researchers that made their genome and metagenomics data available for this type of project to be done. So especially from the Terra Oceans project, but also the Ocean DNA project, which put a lot of this metadata together, and other projects like the GeoTraces project, which really also did a huge amount of metagenomic sequencing and profiling the global ocean. So without these major collecting initiatives and without making their data available, we wouldn't be able to do this project. So we're very grateful for all of those researchers. So thanks so much for listening. I would be remiss not to thank the biology department at the University of Western Ontario that hosted my original visit. This was a re-recording, but the actual visit was a lot of fun and invigorating. And especially a big thanks to Mattia Sanita Lima, who hosted my visit and showed me around campus. It really has an infectious excitement for and enthusiasm for science. So I really appreciated that, and thanks again.